In today's lecture, we're going to talk about clinical trial design. We've been covering a series of topics. Our first lecture was on the background, which included the general history of clinical trials, the regulatory requirements, the ethical requirements, and some of the local requirements. We then went on to cover why do we bother doing clinical trials, what's their purpose in the context of other clinical trial research methodology, such as observational studies or bench research. We also have covered defining the question in a clinical trial, which is sometimes more challenging of the whole enterprise, and how you measure the question once you've defined it. In today's lecture, we're going to talk about basic clinical trial design once you've defined the question and you know the rationale for doing the trial. Just to review briefly, there are several kinds of clinical research. Case reports, that we talked about earlier, which are sometimes and usually anecdotal, but indicate there's a problem. Observational studies are involved in one of three designs. Either we look at patients who have a disease and look backwards to see what the exposure was. We call it case control or retrospective studies. Lung cancer studies, for example, were often of that kind. We talk about cross-sectional studies. We take a snapshot in time. Or a prospective study where you identify a cohort of people and you follow them forward in time. These designs identify potential risk factors with disease. If we get into the drug development or device development, there's certain phases. The early phases, maybe laboratory or bench. You move to phase 1, which might be first in design, or phase 2, where we're trying to find out, does the therapy, the intervention of the device, do what we intended it to do physiologically? And finally, the phase 3 trial, we want to assess, does this intervention have the clinical benefit or effect that we all hope for? In the preclinical trial, we often wind up in the laboratory with either an animal model or some laboratory model. We're looking for dose-response relationships. In phase 1 trials, we're sometimes seeking what's the maximum tolerated dose of the drug that we can give. And sometimes this population is patients who have failed all other therapies. In other cases, it may be a healthy population. But in either case, we're trying to assess a dose-response relationship and how much of the drug or device can you give. In phase 2, we're estimating the drug activity or the device activity, decide if the drug warrants further testing. We want to rule out therapies that aren't working out. Most drugs and some devices fail at either the phase 1 or phase 2 levels. We're also trying to estimate something about the serious toxicity. So in some sense, you can think about this as preliminary data in the development process. The phase 3 trial, which we'll phase most of our attention on, really is to assess the effectiveness of the drug or the therapy. There are various designs which we'll examine, from no control, historical control, a concurrent control, or a randomized control. In all cases, we're trying to test for treatment effect. We sometimes refer to a phase 4 trial, which is a post-phase 3 or a post-approval process where we're looking for safety, long-term safety issues. These trials are sometimes not randomized, but merely a passive assessment of long-term safety issues. The phase 3 trial is where we'll spend our energy. The need for control groups in clinical studies was recognized, but was not really widely accepted until the 1950s. Obviously, if you have dramatic intervention effects, you don't always need a control study. Penicillin, rabies vaccines, examples of that. But that's not the typical case. Generally, we need a proper control group to assess the effect of a new therapy because there's natural history of most diseases and patients vary in how they respond to an intervention. The phase 3 trial is a comparative design, a new experiment, a new intervention versus a control. The challenge is how to establish the control, the historical control, the concurrent control, the randomized control. As you'll see, the randomized control trial is the gold standard, in my opinion, because it's the best method to eliminate several sources of bias, which we must guard against in design of our trial in any experiment we conduct. The purpose of the control group is to allow discrimination of the patient outcomes caused by test intervention from those caused by other factors, the natural progression of the disease, the observer or patient expectations, other treatments. We want it to be a fair comparison if it's going to be informative. In superiority trials, we are testing a controlled treatment may demonstrate efficacy of a test by showing it's better or superior to that of the control. The control might be no treatment, or it might be best standard of care. In any case, we're trying to show that it's better than, superior to, the control. Another class of designs which we'll talk about is what's called the non-inferiority trial. Here, the control trial may demonstrate efficacy by showing that the test treatment is not so dissimilar to the efficacy shown by the known to be effective treatment. The active control, of course, had to be effective under the conditions of the trials in the first place. The new treatment cannot be worse by some pre-specified amount. It's challenging, as we'll see later, as to how to define that pre-specified amount. So two classes of trials, one showing superiority and one showing non-inferiority, not worse than the standard by some amount. In this diagram, I've indicated potential outcomes of a study. What's plotted here is the difference in the event rate between the test drug and the standard drug. If there was no difference, you would have on this graph 0. To the left says that the test drug is better. To the right, it says that the standard drug is better. We've plotted several cases or scenarios of trials. Trial A is a superiority trial because the estimate indicated by the symbol and the line or the whiskers indicating the confidence interval rule out no difference. That is, the difference is certainly greater than some amount, at least a difference of 1%. So we would call it a superiority trial. The next three trials, however, are referred to non-inferiority trial. You notice there's a box labeled zone of non-inferiority. In this trial, we would say that we are willing to accept a new therapy if it's not worse than 2% from the standard. We might accept that because a new therapy is less expensive, less invasive, less toxic, has some advantages, as long as it's not too much worse. So we define the zone of non-inferiority. We say if we can be sure that we're not worse than that, it's okay. Trial B, clearly the upper confidence interval is less than the 2% zone of non-inferiority. So we would say that's clearly a non-inferiority trial, a successful one. Trial C, the estimate is slightly worse, but nevertheless the confidence interval is still less than the upper level of 2%. So we're also happy that that trial, even though slightly worse, it still meets our criteria for non-inferiority. Trial D, however, has an upper confidence interval that exceeds 2%, so it would say it is not non-inferior. In fact, this particular case is not only is it not inferior, it's actually worse, as is trial F. But we want to be sure that we never have the last case where the estimate looks slightly favorable, but the confidence intervals are so wide that we can't conclude anything. We would say that trial is underpowered, not sensitive enough, not large enough, and therefore it would have been unethical to conduct it in the first place. So our goal is to never be in that situation, but this graph was put together to show the difference between superiority and non-inferiority type trials. The use of placebo control. Sometimes this is misunderstood. It might be the case that the patient is getting no treatment plus a placebo tablet, say, but more likely they're getting the standard care plus a placebo. So it's not as though they're not getting any care, they're just not getting any additional care represented by the new intervention. We talk about matched placebos because they're necessary so patients and investigators cannot decode the treatment assignment because patients and investigators have built-in biases. An example is the vitamin C trial for a common cold. A placebo was used, but it was distinguishable. Many individuals that were on the placebo dropped out of the study, and those who knew they were on the vitamin C reported fewer cold symptoms and duration of the cold than those who didn't know. Let's examine the historical control. Here we mean that we're going to compare the new treatment using a series of subjects to a previous series of subjects of comparable risk, as best we can define. It's not randomized, it's not concurrent. It's faster, less expensive, and in fact it may be good for the initial testing of a new treatment. Does it seem like you're doing better? The challenge is, where do you get the historical control data? If you use the literature, it's well known that there's publication bias. That is, not all trials, not all treatments that are evaluated get published in the literature. We're trying to correct that, but still, it's not perfect. So that the publication bias is a factor. If you have a database, the question is it currently available, and how good was it in the first place? So historical control design studies are vulnerable to lots of biases. These may become because there's changes in outcome over time because of the underlying patient population. The criteria for selecting patients, patient care and patient management, peripheral to the treatment, the diagnostic or evaluation criteria may change, and the quality of the data that's available may be problematic. Here's a simple graph. It's not important that you read the fine print, but this represents 3 periods of time where the international definitions of diseases change. So you can see, for example, in the second line, which is ischemic heart disease, there was a change in the definition, and automatically there was a change in the rate. There's other graphs that show this discontinuity when definitions change, and this happens a lot in medicine where our definitions change because of improved technology. It has an impact on the event rates that we see. This graph demonstrates a time trend in death rates for selected causes over a 25-year period of time. You notice that the second line represents cardiovascular disease mortality from 1950 to 1975. Clearly, in the 1970s, the mortality rate for cardiovascular disease was declining dramatically just because of the disease process, the way patients were being treated, and so forth. So if you were comparing a new therapy compared to what you used to do, the 1970s would win over the 1960s, even though perhaps nothing else was changing. This graph illustrates a little more clearly cancer and heart disease deaths. If you're less than 65, for example, you can see that the event rates are changing in heart disease dramatically. They're also changing for cancer mortality, although less dramatically. On the other hand, over 65, you see that heart disease also is declining, whereas cancer death rate is increasing. So the fact that background event rates are changing, using historical controls to compare and evaluate new therapies is problematic because there's not a constancy that you can count on in background event rates. So the historical control studies tend to exaggerate the value of a new treatment. Literature controls are particularly poor. Even historical controls from a previous trial in the same institution or organization can still be problematic. This was demonstrated in the literature where back-to-back studies frequently did not produce the same event rate. One can try to adjust the differences in patient selection, but it's very difficult. Many biases will still remain. In concurrent controls, we're talking about not a randomized study, but where patients are compared, treated by different strategies in the same period. I may compare the patients in one institution to patients in another institution. The advantage, of course, is that, again, it eliminates the time trend. You're talking about the same period of time. And if you work it properly, you can get data of the comparable quality. The disadvantage is the selection bias. Why patients go to one institution versus another is not random. There's a reason, typically. One may be a tertiary referral institution, where one is a primary institution. Treatment groups might not be comparable in those groups. An important thing to remember is that statistical analysis, or sometimes referred to as covariance analysis or covert analysis, is usually not adequate to adjust for these differences. We just do not know all the key variables to measure, to make the adjustments, and even if we did know them, maybe we don't have them. So there's lots of biases in concurrent control, patient selection due to referral patterns, refusals, and subtle differences in eligibility criteria, and then environmental differences, which have to do with diagnosis, supportive care, evaluation methods, data quality, and so forth. So while it's an interesting design, it is not probably adequate for the definitive design that we need. A nice discussion of why the randomized control trial appears in an article by Bayer et al. In 1976, where it talks about patients are assigned at random to either treatment or control, and the randomization process is considered to be the gold standard. The advantages of the randomized control trial are as shown here. Randomization tends to produce comparable groups. In the randomized trial, you're always subject to chance. You can't get away from that. In the concurrent trial, you're subject to chance and selection bias. The historical control trial, you're subject to chance, selection bias, and time trends or time biases. So the randomized trial gets rid of most of the biases we can think about, and we're left to chance, which we can control to some extent by the size of the study and the design we use. A second benefit, often not appreciated, is that the randomization process produces valid statistical tests so that we don't have to make any assumptions about distributions and so forth. There are some disadvantages to the randomized control trial. The subjects may not represent the general population. There is the volunteer effect. You have to volunteer to be put into the randomized trial. It takes twice as many patients because you have a treatment group and a control group. And some people do not accept the randomization process and may choose not to enter into the trial, which is their right. And it does add some administrative complexity and overhead. Nevertheless, we believe that the randomized trial is still the standard because of its impact on reducing bias. This is a study that was published in 1979 by Richard Pito. He's looking at trials of anticoagulant therapy and observing their effect on mortality. In searching the literature, which is not always perfect, identify 18 studies which use historical control comparisons, 900 patients, and in 15 of the 18 studies, the observed effect was significant at the p less than 0.05 level, and roughly the observed effect was about a 50% reduction in mortality. In the 8 concurrent studies that he could identify, that is, you're comparing new therapies in one place to a therapy in another place, 3,000 patients, In 5 of those 8 studies, the result was significant, p less than 0.05, again suggesting about a 50% effect. For the 6 randomized studies he could identify, again with 3,000 patients, only 1 of those 6 was significant, and the effect was much less, that is 20%. So while it looks as though the therapy has some potential benefits, the estimate of the effect is definitely increased in the historical control and the concurrent control design because of the biases that exist in the designs. So we believe that randomized controlled trial is the best, most ethical way to conduct a study and find out which treatment is best. It reduces, in fact, the risk of being on the inferior treatment, and certainly reduces the risk for future patients. An interesting example about randomization is demonstrated by an article by Silverman in 1977, which examines the practice of using high-dose oxygen in premature infants. It was common practice. Over time, there began to be a suspicion about the frequency of blindness in these premature infants, and later a trial showed that the high dose, in fact, caused the blindness. In the randomized phase 3 trial, we assume that patients are enrolled, have satisfied the eligibility criteria, and given consent, and then we're going to use a balanced randomization that each treatment will get the same number of patients. So that's not necessary, but for our purposes, we'll assume to put the same number of patients in each the treatment group and the control group. There are different experimental designs we can use to answer these questions, however. Again, choices to be made. There are several commonly used designs we can use to answer the phase 3 question. I've listed several of them. We will use our time to discuss a couple of them. The most common design is the parallel randomized trial. Patients evaluate for their eligibility. If they're not eligible, they're out. If they're eligible and don't consent, we are also taken out of the study. But if they're eligible and consent, we randomize them to A versus B. We will then compare patients on treatment A to the patients on treatment B for their benefits in their primary question, their primary metrics, their adverse events, and other such outcomes. Examples of these designs are numerous. A trial in heart failure with using the drug risnerenone. We've talked a little bit about the cardiac arrhythmia suppression trial, where we evaluated antiarrhythmic drugs in patients with experiencing arrhythmias. The diabetes complications control trial, looking at various ways to control glucose levels compared to a standard approach. We've already talked about the nocturnal oxygen therapy trial, high-dose oxygen or continuous oxygen versus 12 hours of oxygen. And we alluded to the intermittent positive pressure breathing trial, a device to deliver a bronchodilator under pressure versus a handheld nebulizer. This design is the workhorse of clinical trials and is the most frequent design that you will see. Some trials have a little run-in period where we know that non-compliance to therapy by the patient may seriously impair our ability to detect the effect of the intervention. So one solution, at least in drug trials, is to assign all patients for some period of time to a placebo, be taken over a brief period, and see if the patient is able to comply with taking the therapy, the pill, complying with the procedures, the measurements, and so forth. We often call this the run-in period. You can also use an active drug in that same period. Either one is permissible. Your goal is to find out whether patients will comply with the procedures that are necessary. So then the run-in design, as depicted in this diagram, you would still have your screening process, your consent process, and then there's a run-in period. If the patient doesn't perform satisfactorily, you may choose not to randomize them. If they conduct themselves in the run-in period to the satisfactory meet the requirements of the trial, or the protocol, then you would randomize them to A versus B, A or B, and compare the two effects of the treatment, A versus B. Again, a sort of parallel design with a run-in period ahead of it. Examples of run-in trials. We've already talked about the cardiac arrhythmia suppression trial, had a run-in period. The diabetes control and complications trial had a run-in period. And the physician's health study, a trial which we've not yet discussed, was a trial of aspirin versus placebo, and it also had a run-in period as whether patients could tolerate taking aspirin without having any GI or other kinds of bleeding problem. So it's a very effective design to improve compliance. Another design not used very often is the withdrawal study. Imagine you have a therapy, say a beta blocker following a heart attack, and you know it's effective in reducing mortality over some period of time following that heart attack. Question is, how long should you continue the beta blocker? So one way to examine that is to, at some point in time, 2 years, 3 years, randomize the patients to either keep taking the drug or to stop taking the drug. The advantage of this design is you have access to patients that are already in your trial, and you know that they are complying because you have that experience. And so you can ask the question, is there continued therapeutic benefit? Of course, there's a disadvantage, and this is a selected population. It's not population in your clinic, but those who have been screened to be eligible, who've consented, and have gone a certain period of time without an event that took them out of the study. The disease process itself may have changed, but it does give us the best evidence about how long should therapy continued. Another class of designs is referred to as the cluster randomization design. In this study, the groups of clinics or communities are randomized to treatment or control. It might be a study examining the impact on a community of fluoridation of the water, or a breast self-examination program in different clinics, or a smoking cessation intervention in school children. The advantages are sometimes that's the only logistical way you can do this study. If you're going to intervene on children, for example, in a school, it's unlikely you can randomize the individual children. More likely you can randomize classrooms or the school itself. It avoids contamination. People in the school or the classroom are doing the same thing. It also sometimes a good design if you want to do mass intervention in public health arena. The disadvantage is that sometimes the effective sample size is less than you think. It's less than the number of subjects because within a classroom, the school, they're doing the same thing. So that it's not as though you've got independent replications, but there's a correlation. So we will see later that we have to adjust our sample size estimation to take into account that clustering. But nevertheless, it's a very effective design for certain kinds of studies where you're intervening on a mass scale. The crossover design is an interesting design where you take a treatment and each individual, each subject gets both treatment but in different order. I've depicted here a little diagram. We can imagine a first period and a second period, and you have two treatments, A and B. So in the first group, patients are randomized to get treatment A first and then get treatment B. The second group is randomized in the reverse order. First get treatment B and then treatment A. The advantages of this design is that each patient serves as their own control, and thus you wind up with a smaller sample size. The disadvantages, however, can be used in every setting. It certainly can't be used in the acute disease setting. You assume that the disease must be quite stable or reasonably stable. But the most important assumption in this design is that there's no carryover effect. That is, the therapy that's applied in the first period will not have any impact on the therapy applied in the second period. In the extreme case, if the therapy in period 1 cured the disease, then in the second period there's nothing for that therapy to do. That's unlikely to be the case, but nevertheless it indicates this assumption of carryover. If it turns out you do this design and discover there's a carryover effect, you wind up having essentially a study that's half as big as you thought. Those who got treatment A first, treatment B second, or the other, but you can't combine the two strategies. A little more complicated version of this is the so-called factorial design. Here, we're going to try to get two studies for the price of one. We have factor 1 and factor 2. Factor 1 might be incidence of cancer. Factor 2 might be incidence of coronary heart disease. We have two therapies, treatment A and treatment B. So we're going to randomize subjects to one of 4 cells. They each get placebo. They get placebo and a treatment B. They get a placebo and treatment A. Or they get both treatment A and treatment B. So we have these 4 cells, and what we're interested in is averaging all the patients who got treatment B compared to the placebo to evaluate, in this case, factor 1, or compare all patients who got A versus those who got placebo on factor 2. So we get 2 comparisons, A versus placebo and B versus placebo, in one population. So yes, there's some advantages. 2 studies for the price of 1. We may, in some cases, discover interactions we didn't know about, and that may be, in fact, quite important. However, interactions might, in fact, hinder our trial to detect the main effect. It's difficult to detect interactions, as we mentioned earlier. And something that I worry about is any chance that the compliance to treatment A might have some impact on compliance to treatment B. That is, if one therapy is quite toxic, patients may just say, forget about it, I don't want to participate in this trial anymore. And so you not only lose your ability to compare treatment A, but you lose treatment B as well. There are many examples of factorial designs. The Physician's Health Study, which I mentioned earlier, compared aspirin versus placebo and beta-carotene versus placebo. The second component of the Physician's Health Study was the beta-carotene arm. This article, published in 1996, shows that the use of beta-carotene at high doses did not, in fact, reduce the incidence of cancer, but, in fact, increased the incidence of cancer. This is in contrast to the observational data, which suggested that low values of serum beta-carotene would increase the incidence of cancer, suggesting that if you increase serum beta-carotene, one should get a beneficial effect. In this case, there was absolutely no impact. The relative risk was 1, essentially. And now I want to examine a little bit of the issue of superiority design versus non-inferiority. We already said that the superiority design is to show that the new treatment is better than the control or the standard, and that non-inferiority wants to show that the new treatment is not worse than the standard by some margin, and that it would have beaten placebo, in fact, if placebo arm had been there, being somewhat of a regulatory requirement. Non-inferiority trials have some specific challenges. It requires a very high-quality trial because poor execution of a trial would favor non-inferiority. So the challenge is you must do the best job you've ever done. It also requires using a strong control. A weak control will also favor non-inferiority. The treatment margin is somewhat arbitrary. It also requires high power and often large sample size. And finally, we try to impute the treatment effect versus placebo by using historical control data. The problem is that the historical control data, as we've already said, may not be so relevant. Things have changed. There's not a constancy. And therefore, the imputed estimate of the treatment versus placebo is not a very robust estimate. We've already gone over this slide, but just to draw your attention again to the three non-inferiority cases listed here, trials B, C, and D. First one definitely being non-inferior, that the upper confidence interval is less than the 2% margin of indifference. The second trial is slightly worse by its estimate, still meets the non-inferiority criteria. And trial D definitely does not meet the non-inferiority criteria because it includes a difference of 2% as a possibility. And trial E, of course, is not inferior, but in fact inferior. An example of a non-inferior trial is a trial called Optimal, which was published in Lancet in the year 2002. We know that ACE inhibitors reduce mortality in high-risk patients following a heart attack. There are selective angiotensin II receptors, or so-called ARBs, that may be an alternative because of complete blockade ability and better tolerable. The hypothesis was, with a new drug, losartan, superior, hopefully, or at least non-inferior to captopril, the standard, in decreasing all-cause mortality in patients who had high risk following myocardial infarction. The study design was double-blind, randomized, placebo-controlled. It was multi-center, and the primary outcome was all-cause mortality, and a design which we'll talk about is event-driven, where we had a target of 937 deaths to achieve the sensitivity or power. Captopril has well-documented benefits. It's the standard. It was going to be used in a standard dose. It's a drug that's widely available, in fact, even available as a generic at the time of this trial. The reason for the 937 deaths was that would give us 95% power to detect a 20% difference, issues which we'll talk about in more detail in the sample size lectures. But in this study, the non-inferiority margin was determined to be 10% on a relative scale, chosen based on other trials, previous trials, placebo-controlled trials of the ACE inhibitor study. And it was going to use a meta-analysis by intention to treat, which we will again talk about in the later lectures. Well, here are the results of that study. The relative risk between losartan and captopril was 1.13, favoring captopril, the standard. As the new therapy was, yes, worse. In fact, it was worse by more than was set forth in the margin of indifference, which was 10%. So this trial, while it was well conducted with a new therapy, did not meet the criteria of non-inferiority. You can also see that if you look at subgroups, the inferiority of losartan was pretty consistent across a wide range of classic heart failure risk factors. So it's not as though there was one population benefiting and the other one was not benefiting essentially anywhere. To illustrate the imputation aspect, I've listed here three relative risks. The first one is the one we just looked at, the comparison of losartan to captopril, where the relative risk was 1.126. Historically, based on literature, trials which compared captopril, the standard versus placebo, indicated a relative risk of 0.8. There's a reduction of 20% almost in the mortality rate. If you multiply the relative risk of losartan versus captopril as the first comparison, times the relative risk of captopril versus placebo, algebraically you get an estimate of losartan versus placebo, which is 0.906. So you can get an imputation about, yes, losartan would have beaten placebo had placebo been here, even though it is inferior, or at least doesn't meet the non-inferiority criteria compared to captopril. But to do this calculation requires a very strong assumption that everything is constant, because the historical data is still relevant today. Very strong assumption. We also talk about large, simple trials. Many trials were looking for small benefits, but because of the large number of people at risk, still have a very important public health benefit. So even if we want to look at for modest benefits, which would implement in large populations, we need large studies. So in order to do these, we have to really be very specific and very simple due to costs. And so we must, in fact, limit the amount of data that we're going to collect and pick outcomes which are easily ascertained and very reliable, not subject to bias. An example of this kind of design is the Capri trial, which was again published in Lancet in 1996, looking at a population which had ischemic strokes, myocardial infarction, comparing clopidogrel versus aspirin. Here you can see that 9,577 patients were recruited to one arm and 9,566 to the other arm. Very large numbers. Do this trial be very simple. The results, by the way, suggested clopidogrel did better on ischemic stroke, myocardial infarction, and vascular death. At least the results are in the favorable direction and overall significant. We also will talk later, but just to mention now, the idea of a sequential trial. The idea here is you continue to randomize subjects or continue to follow them until your null hypothesis of no difference is either rejected or accepted. There's a large statistical literature for classical sequential designs, which were developed for the industrial setting, manufacturing setting. But those methods were modified to apply to clinical trials. An example of the use of a sequential design is in the beta blocker heart attack trial, which has been published in the American Medical Association journal. Mortality was the outcome. It was randomized, double-blind, placebo-controlled. 3,837 men and women were randomized to a drug, propranolol, or a placebo. What I've depicted here is the essence of the sequential design. What's plotted here is calendar time from late 1978 up to 1982 on the horizontal axis. On the vertical axis is the standardized test statistic. The dashed line represents the level of the test statistic that would be typically associated with a p-value of less than 0.05. That test statistic would be 1.96 or larger. What's plotted then for each period in time is the test statistic as we saw it in real time. As you can see, the plot crosses the dashed line of a nominal p-value of 0.05, and the trial continues on until it crosses the solid line. The dashed line is sort of the nominal p.05 value, and we know that if we keep looking at data, and we'll discuss this a lot more in a later lecture, that you can by chance alone find results and make false claims. The solid line is a boundary that says if you want to reduce your chances of false claims, you must have very strong evidence early before you're willing to quit. As you can see, in late 1981, we crossed the boundary and the trial was terminated. There are many issues which we'll discuss later about reasons for how this trial was terminated, but for right now, it's an example of a sequential study that was launched and stopped early because of effects, in this case, beneficial effects. Another class of trials is the therapeutic versus prevention trials. In prevention trials, we're either trying to prevent the disease from occurring in the first place, we sometimes refer to that as primary prevention trials, or secondary prevention trials where the disease has occurred and we're trying to prevent it again from occurring. For example, a patient having a heart attack, trying to prevent the second heart attack, or worse, fatal heart attack. In a therapeutic trial, we're trying to intervene on the disease in an acute setting. A cancer patient being diagnosed, we want to take care of the immediate cancer risk. A patient coming into a coronary care unit with severe angina, we want to take care of the pending heart attack. There are many differences in these trials in terms of recruitment strategies. For example, the therapeutic trials, patients come to you. In prevention trials, you typically have to find them. Nevertheless, the basic fundamentals apply to therapeutic trials and prevention trials. Yes, there are differences in complexity, recruitment strategies, compliance, length of follow-up, and perhaps size of the trial. But the fundamentals that we examine here in these lectures will apply to both. We talk a little bit about masking or blinding. The no blind or unblinded trial is that all patients know the treatment. Everyone knows the treatment, essentially. In the single blind, the patient does not know, but the healthcare team does. In the double blind, neither patient nor healthcare provider know the treatment. Sometimes people talk about the triple blind, when neither patient or physician or those who monitor the trial know the treatment. As we'll talk about in the monitoring lectures, the double blind is recommended. Triple blind, I believe, is not a good approach. Why do we bother masking? Well, there's lots of biases that come into a trial. Treatment decisions can be biased by the knowledge of the treatment that's being given, especially the treatment team has a perceived concept about which treatment is best. It can affect dose modifications, it can affect the intensity of patient examinations or a need for additional treatments, and it can even influence the patient's attitude towards their enthusiasm for one of the therapies. We mask evaluators because sometimes endpoints are subjective. The evaluator bias can lead to more favorable response on one treatment or the other. Even supposedly hard endpoints often require some clinical judgment. A little example about the impact of bias of an investigator. This is a study which actually I participated in, part of it, was a taste and smell study. It was published in 1972 and also in 1976. In this case, the investigator, Dr. Henkin, observed that patients who had taste or smell abnormalities had low serum zinc. So his approach was to increase serum zinc and evaluate the patient's success on taste and smell. He initially designed a single-blind study that the patients did not know whether they were getting the zinc or the placebo tablet. He knew as he administered the tablets. He devised a very clever system of 8 modalities of taste and smell. And in his single-blind trial, all 8 of the modalities improved patients treated with zinc, and none improved on placebo. When he completed that trial, his superiors suggested he repeat the trial in a double-blind fashion. In the double-blind fashion, only 5 of the 8 modalities improved on zinc. In fact, 7 of the 8 modalities improved on placebo. Clearly, his enthusiasm and knowledge of the treatment being assigned impacted his assessment of the patient's ability to taste and smell. So to summarize what we discussed in this lecture on the design of clinical trials, we have many options that we can choose from. The design we choose should be linked closely to the questions being asked, measurements that are being used. Each design has their advantages and disadvantages, but in general, the randomized controlled trial, regardless of which design option you use, will prove to be superior to the unrandomized, largely because it controls and eliminates most of the biases that we have to deal with. In the next lecture, we will be talking about sample size. As we discussed, once you've defined the question and the metric and the design, the next challenge will be how many subjects do I need in my study to achieve the goals I've set before me. So we'll examine the issues, the components of determining the right sample size for your study.